Lecture 4, How Genes Work, Part 3. This is the last of a three-part video series for Lecture 4. What we're going to talk about a little bit more is how to use or apply the genetic code, and we're going to talk about mutations. Alright, so we just finished looking at the standard codon table, which shows us the standard RNA codons for the 20 common amino acids. Now let's think about how to use it. So looking at the example on the left, the red is DNA, the yellow is messenger RNA, and the green is a polypeptide. So DNA occurs in cells as two strands. It's a double-stranded molecule. Only one of those two strands gets transcribed. That's important to remember. So in this case, the authors of the text are telling us that it is the bottom strand. There are ways of figuring that out, but we're not going to get into that right now. So the bottom strand is transcribed by RNA polymerase to make this messenger RNA molecule. So it's following, the RNA polymerase is following the DNA to RNA conversion or base pairing rules. So where there is a T in the DNA, there is an A in the RNA. Over here you can see that. Where there is an A in the DNA, there is a U in the RNA. Where there is a C in the DNA, there is a G. Where there is a G in the DNA, there is a C in the RNA. So we're using the DNA to RNA base pairing rules that are important in transcription, as explained in the previous part of this lecture. That AUG signifies methionine, which is also the start codon, and MET stands for methionine. That codon GCC signifies the amino acid alanine. Okay, so this is a polypeptide. It's a series of amino acids connected by peptide bonds. The UAA is a stop codon, and that's where the polypeptide ends. So this step here is transcription. This step here is translation. Transcription is done by RNA polymerase, translation by the ribosome. Now I'd like you to take a stab at transcribing and translating this sequence of DNA as shown here. Let's think about how you would do that. Alright, so let's focus on the top left. So we're going to transcribe this sequence. And I'm not going to do the whole thing. I'm just going to do the first one or two codons. So here is our sequence of DNA. It is T, A, C. The corresponding RNA, if we use the rules I talked about, where there's a T in the DNA, there's going to be an A in the RNA. Where there's an A in the DNA, there's going to be a U in the RNA. Where there's a C in the DNA, there's going to be a G in the RNA. Now let's translate that RNA codon. And if we look for that RNA codon, what we'll see is it corresponds to methionine. Okay? So that's the process of translation, going from the RNA to the amino acid. You should do this for practice and fill in what belongs in each of these areas, and you can check your answer using the key in your textbook for this chapter. It's chapter 16. Alright, let's talk a little bit about what happens when there are changes in an organism's DNA. This is something that can happen pretty easily during DNA replication, when you copy the DNA, which you do whenever you want to copy the chromosomes, so you do that whenever you want to undergo cell division. A mutation is a permanent change in an organism's DNA. A lot of errors happen during DNA replication, and when those errors happen, if they're not corrected, they become fixed. And the result of that permanent change, is a mutation. If you have a mutation in germ cells, so those are the cells that give rise to sperm and egg, for example, in a mammal such as ourselves, then it's inherited. Change. If you have mutation that occurs in somatic cells, then it's not inherited, but you have something like a tumor or cancer. Because of the way that DNA replication occurs, which is something that we're going to talk about later this semester, it occurs using a process that is semi-conservative. That means that when you have two molecules formed from one, half the molecule will be from the original piece shown here, and the other half will be new. That's the reason that only one of these two molecules is altered. Let's think a little bit about the different kinds of mutations that can occur and what results they can have on cells. Okay, let's talk about point mutations. Point mutations are changes in genetic sequence that affect one or only a few nucleotides. Typically, therefore, the change occurs or is something that affects just one gene or perhaps the regulatory region for just one single gene. These can be very tiny changes, but they can have profound impacts. Before we go through the terminology and the information in this table, I want to point out that mutations can be beneficial, they can be neutral, or they can be deleterious with regards to their consequence to the organism. A beneficial mutation might be a mutation that changes a protein so that it works better in the organism and actually contributes to increased overall fitness or ability to reproduce and therefore transmit one's genes to that organism. A neutral mutation is a mutation that has really no detectable change to the phenotype of the organism and therefore its fitness. So that might mean that you had a change in nucleotide sequence but yet no real change in the functional protein, that it works about the same. A deleterious mutation is one that has a harmful effect on the phenotype of the organism and therefore a harmful effect on the organism's fitness. So that might mean that you lost a protein entirely or that the protein was less functional and therefore the phenotype influenced by that protein was harmed. Mutations can be silent, which are changes in nucleotide sequence that do not change the amino acid specified by a codon. To understand that, you need to think back to the fact that the genetic code is redundant. So there's more than one codon for most of the amino acids. So here we have a genetic change for one codon to another, but they both encode tyrosine. So that would result, of course, in no change in the protein, therefore no change in the phenotype, no impact on the organism's fitness. A missense mutation means that the nucleotide sequence has changed and in such a way that the amino acid is now different. So here is a missense mutation. We've gone from having the amino acid tryptophan to the amino acid cysteine at that position. The effect of that on the organism is hard to predict. You have to look at it in the overall context of what amino acid has been changed, what it becomes, and how it affects the protein that it is found within. So it may be beneficial, neutral, or deleterious. It just depends. A nonsense mutation means that you have a change in nucleotide sequence such that you go from having an amino acid at one position to having a stop codon at that position, and that's what's shown here. We go from having a tryptophan to a stop codon, and then notice that you have nothing. So now we've got a prematurely truncated protein. The messenger RNA is not transcribed downstream of that position. This is not used, and so there's no corresponding polypeptide for that position. That's usually a deleterious mutation. A frame shift mutation occurs when we add or delete one or perhaps two nucleotides. That's what's happened here. Cytosine has been inserted. As a result, that codon is now encoding serine instead of tryptophan, and everything downstream of that mutation has been bumped. So in fact, every codon is different and now encodes a different suite of amino acids than in the original protein. These tend to be pretty serious mutations, generally deleterious, because they can completely change the protein. However, from an evolutionary perspective, this is actually a way that you can get novel or new proteins evolving and forming in an organism. Other types of mutations can be much, much larger. So the category of mutation known as a chromosomal mutation is what happens when you have a large-scale change in the content or number of chromosomes. So you have one too many or one too few chromosomes, or you have large chunks or regions of one chromosome moving to another chromosome, something called transposition. This picture that you're looking at is called a spectral karyotype. Spectral because it's done using a kind of spectroscopy, which is a way of imaging color, and karyotype because it's essentially a fingerprint of all the chromosomes in a cell. And this is the karyotype of a breast cancer cell. Cancer cells have very unusual numbers of chromosomes and altered chromosomal content. Unusual numbers of chromosomes is referred to as aneuploidy. So if this was a normal, healthy cell, and it was a human cell, what you would see would be 23 matched chromosomes. If it was a female from a female individual, you would see identical XXs. If it was a male, you would see one X and then a much shorter, smaller Y chromosome. So they wouldn't be matched, but all the other chromosomes would be matched with regards to length. There would be two of them, and they would be all the same color because they would have the same basic composition. Here we see something radically different. Chromosome number 2 over here has in fact 3 of them, and none of them are the same. That's really not right. And it's something you see very commonly in cancer cells. They're severely disrupted. So with that, I'm going to complete this part of the lecture.